Okay, so I think we will get started this morning, this afternoon, this morning, depending on where you are joining us. I would like to commence our formal proceedings this morning by acknowledging the Gadigal people as the traditional custodians of the unceded land on which our symposium, our seminar takes place today. And as this is a hybrid meeting, I'd also like to acknowledge the traditional custodians of the many lands from which our virtual participants are joining us. We pay our respects to their elders, past, present, and emerging, and recognise their cultural and spiritual relationships with the lands, waters, and seas. It is our privilege to learn from their rich contributions to both society and science as we work towards finding more effective ways to help all children with cancer. I extend that same respect to Aboriginal and Torres Strait Islander peoples and colleagues here today. So welcome everybody. Thank you to all of you from around Australia and elsewhere for joining our first seminar, Zero seminar for 2023. We're very grateful for the participation and attendance of so many of you today. I'd like to warmly welcome today's speaker, Associate Professor Mark Cowley, who is the Child Cancer Computational Biology Group Leader at Children's Cancer Institute here in Sydney. He's also a co-leader of the ACRF Child Cancer Liquid Biopsy Program and Genome Informatics Leader for the Zero Childhood Cancer Program. And Mark is also the co-leader of the next generation of the Luminesce Alliance Data Enabling Platform. As you can imagine, he's had many years of expertise and experience in genomics and precision medicine, and having previously spent 11 years, I think it is, at the Garvan Institute when he was appointed as a program leader of translational genomics, informatics, and cancer genomics. So during that time, he generated a range of bioinformatics methods that enabled not only most, but eventually Zero, as well as the clinically accredited whole genome analysis test that was applied to many Australian patients during his time at the Garvan. So Mark's been with CCI, with the Children's Cancer Institute, since 2018 officially, although we were definitely leveraging him for a few years before that. And he's leading the computational biology group, as I said. He is here to talk to you today about our emerging data platform strategies and various other very interesting items. A reminder to all of you that if you have any questions, to please feel free to pop them in the chat, and we should have some question time at the end. So Mark, over to you. Thank you.
Thank you, Ness, for that overwhelmingly warm introduction. Yeah, there's reminders of good times in the past for sure. Let me try and share this. Okay, how's that? Can you see that?
Yeah, you're good to go, Mark.
All right, cool. Thank you, Ness. Yes, thank you so much for the chance to be here. I wanted to do something different today. I just gave a talk at Lawn Cancer, which was focused on the science and the expansion and really that focus on closing the gap for these children with high-risk cancer and increasingly now for children with non-high-risk cancer. I think I've given a talk like this to various different audiences, but never really the domestic one. So I've had the chance to show some of this to the CBTN group and the Profile group in Canada. So what I really wanted to do today is, normally I like to talk about what we've done, but there's lots of things that we're doing that I think is high time to tell you all and bring you on a journey and let you know some of the thinking of the things that we're trying to build. So that's really a way to frame that. If you go to zerochildhoodcancer.cloud today, you won't see anything because it's still an initiative that we're building. And so some of this is aspirational and some of this is what we've already been building. All right, so with that starting, I would love to jump into the most important slide of the deck, which is to thank the Computational Biology Group at Children's Cancer Institute, who really a remarkable group of scientists, lots of engineers, designers, students, who really helped make this happen. And I'm so grateful to them. I'm not sure they've even heard me say what I'm about to say in this way stitched together. So hopefully that's useful. And just to remind you that if you don't know me or what we do, we're traditionally computational biologists, and we use the tools of data science, data engineering, software engineering, and genome and transcriptome informatics to try and use data to make inroads into tackling childhood cancer. And as Ness has pointed out, that journey to get to the Children's Cancer Institute flowed through adult cancer analysis and children with rare genetic disease. So that, I guess, that collective experience of this group really comes through in the approaches that we try and take now. So I'm going to revisit this slide because I'm going to try and go broad and not too deep, but just to try and give you some anchor points as to what we're talking about today. I really want to start with a reminder of what 02 is and some of the challenges that we've been thinking about for scaling that up. Then into the data vision for the whole program, some historical perspective on what we built, why and when, just to give you some understanding of kind of where we are at in our journey. We'd love to have built all the solutions already, but we've had to be judicious and solve different problems in a different order. Then I'd like to tell you about the cloud, this ambition to build a digital ecosystem with a deeper dive into two of the most mature parts of that solution, the zero dash and the MTB notes. And then really end with new, largely unfunded opportunities that we think are really important, are in progress, and really want to bring you on the journey as to what we're trying to do there. Okay, so 02. I realize we have a fairly broad audience, so I just wanted to put this placeholder in to say that we are opening 02 right now at all 9 sites across the country, ambitions to open at the 2 sites in New Zealand. And essentially what we're doing is the same comprehensive molecular profiling that we've been doing in the PRISM study. So that is deep tumor whole genome sequencing, RNA analysis and methylome analysis. All high risk and rare cancers are eligible just like they were in PRISM. And the focus there will still be on finding the diagnosis and the best treatment options for those patients. Where possible, we'll do drug screening and animal modeling. And if the patient unfortunately relapses or progresses, we'll rebiopsy them and repeat the molecular analysis, see what's changed, see if there's anything new to report. What's new is that now all non-high-risk cancers will be eligible. Here, the focus is less on treatment, but more on getting the diagnosis and predicting the prognosis rapidly. And we're doing this in a phased approach. So we're first opening all brain tumors, and then likely neuroblastomas next. And then after that, by the end of the year, we should have opened up all disease streams. So that's just a way to manage the scale up. One of the ways we're doing this is to create a virtual gene panel for each disease stream. And so the wonderful curation team will focus their molecular interpretation on those genes in those panels. But if we don't find anything, all the data still is there, so we can revisit. And we can still, of course, do lots of discovery research on the rest of the data too. The goal is a semi-automated molecular report in 4 weeks, actually, aspirationally 3. This slide was made a little while ago. Yes, I'm going to touch on the other stuff later, but I see we've got colleagues on the line with interest in the psychosocial components, the health economics, germline analysis, and e-consent studies. So there's lots of additional things beyond my view of what 02 is. So from a data science perspective, 02 is going to be a data challenge. Here we're showing quarterly growth in the number of new patients recruited and the total number of patients, potentially getting up to about 4,000 patients by the end of the study. That means huge growth in data storage, and that means huge growth in the amount of compute that we need to do to analyze all of that data. So this slide's occupied our brains for a couple of years. I'm not going to go deeply into some of the analytical solutions to this, but suffice to say, we've been building new analysis pipelines, new tools that make this more optimal, new data compression stuff that I won't go into, just to try and tackle this aspect of 02's growth. And we've been working closely with our TSG group to build new data storage systems as well. So I want to spend 3 slides to frame some of the vision of what we have for 0 and the data that we're collecting. This first vision is a little bit inward facing, and the next 2 are more outward facing. But really the idea is that we are already collecting and want to build a data system where we can harmonize all of the DNA and RNA sequencing data, the epigenetic data that we're collecting, obtain clinical data, as I'll end the talk, talking about how we might get some of that clinical data from the electronic medical record systems so that we can get better clinical data. Histopathology, cytogenetics, molecular genetics, imaging, laboratory tests, outcomes. This is a really very difficult part of the data collection that we currently do quite manually, like most other precision medicine groups like this. Also keeping track of all the cancer information, the different cancer types that we're seeing, characteristics of those, all the genetic variants that we discover. We're over 100 million genetic variants that we've seen throughout the last phase of zero, storing all of that information, increasingly bringing on the liquid biopsy program, which I probably won't talk about much today, but storing that data and how that relates to how tumors change over time. All the drug screening results that we've been creating with many of you on the line, high throughput drug screening, CRISPR screens eventually, and patient-derived xenografts. This one's aspirational. We don't yet have medical imaging data in here, but it would be great to one day. Information about trials and drugs. So we want to build data systems that track all of this. It's not just genomics data. I'm calling this precision medicine data ecosystem because it's got clinical data, outcome data, drugs, and not just genotypes, which a lot of others, if you Google genomics data lake, you might see others storing genomic data only. The other part is we increasingly see the role for zero as enabling national and international research. So we already support lots of pediatric precision medicine programs like the one from Hudson, working with the team at ProCan. Not really sure why I'm specifically calling out Deb and Matt, but just to say that they have exhaustive research programs already in RNA sequencing of leukemias and childhood brain tumors, and that we are supporting those researchers and many others with providing access to data. We're working very closely with the disease stream groups through 02, which helps to have them in the driver's seat to define the research questions, and collaborating with the Canadian version of Zero, which is called Profile, and specifically with the Sick Kids group in Toronto, and that we've submitted and approved 25 data access requests nationally since 2019. We see an increasing role for the Zero data supporting registries that exist in the country. So we're already trying to get conversations started about the thyroid registry, but that idea that we can collect the data at once and then work with other registries to have their registries populated is something front of mind for us. Likewise for tumor banks, all of the tumors that we sequence end up being submitted by you all around the country and into the CCI tumor bank, and in turn into the ANSTOG tumor bank. So we see a role for Zero's data collection to support those biobanks. And we already support the inclusion of patients enrolling onto different clinical trials, such as Inform2 and Logic and SIOPEN. So that's a goal for ours, to increase the utility of what we're collecting to enable patients to get onto treatment trials and other researchers to do their research. And the third piece of the vision is that we'd really like to be able to connect our data to data around the world. So there are groups, say at Children's Hospital of Philadelphia, the CBTN group, or St. Jude, or the Heidelberg group, or even groups around Australia. And we'd love to be able to build data systems that help find patients. For instance, we think of this like the Expedia model, where you might want to find me data from all patients with a specific diagnosis, a specific age, with a specific mutation and a specific survival. Sounds reasonably easy to write, but devilishly difficult to solve computationally. So I'm going to touch on this later on in the talk as well. But if you were to run a query in a data system, ideally you'd get a result that says, hey, we found lots of patients, some from Zero, some from Philadelphia, some from St. Jude, some from Inform. And then that research would be able to put in a data access request to get that data, do their research. We've been partnering with the Australian Biocommons to enable this, and I'll talk more about that later. I just really wanted to frame the talk today with that kind of vision, that we realize we've got a unique data asset and we're working very hard to work out how to make it more usable by the world. There's a bit of a timeline of what we've built and when, and a little bit about why. So if we cast our mind back to 2015, Personally, we'd been analysing whole genomes at that stage for about 3 or 4 years, but we needed to focus and optimise those tools for childhood cancer specifically. But once we had that working through Target, we didn't touch that too much and then went through another push to re-optimize the tools and make sure our false positive rates are low and we're not missing things. Back then, the pipeline cost about $200 per tumor to run. We soon did the modeling on all of those tumors that I told you about in 02. And so we've worked with the CHOP group to bring the Cavatica platform locally and work out how to get that pipeline analysis down to $50 through some a huge amount of engineering that I'm not going to talk about today, but clearly that's going to help us scale up and help us retrospectively analyze the data. We focus a lot, unlike genomicists that do discovery research purely, we focus a lot on building tools that help us interpret that genomic data really fast. And so our amazing curation team can interpret it, and we can put that information back in the hands of the oncologist quite quickly, or the pathologist. So we first built a system called Sieve, primarily for rare disease analysis, but we adapted it to cancer. And then we rapidly realized childhood cancer needed a different solution. So Marie really spearheaded the development of GLUI, which glues together lots of different information together. And we've just done a massive re-engineering of Zero Dash, which I'm going to show you about, again, for optimization. We've also built a knowledge base, which I'm going to talk to you about later on. We really wanted to get better quality clinical data this time around. So we had a huge effort of many people at many sites as well on developing a great data dictionary and new data capture systems. So that's all been going on, and you can kind of tell the density of activity that's been happening around here to get 02 rolled out. Oops. We've been thinking about building data sharing portals for a long time. We partnered with Biocommons to trial a technology called Gen 3, which, to be honest, didn't really work out for us. So if that had have worked out, we would already have a data sharing portal live and online for you to use. But I guess not every project and technology we back works out. So we pivoted from that to work on this new Beacon V2, which I'm going to talk to you about later. And then these are the initiatives that we're working on, again, which I'm going to talk to you about later. I'm getting a bit sick of saying that, but just kind of wanted to frame what we've been building and why. Early on, we had to analyze the genome data as well as we could. Now we've refined it. Now we focused on clinical data. And there's a huge focus now on building systems to share this data with others. I noticed there's a couple of colleagues on the line from Minderoo, and they have a new federated cancer data initiative, which is something we're working with them on, which is how we can get high quality molecular and clinical data with a multi-institution organization website, essentially, to be able to access data from 3 different sites. This is a very wordy version of that. I don't think I'm going to go into that at all. I wanted to point out that after having built all of those different data systems, these are the types of data assets that we have in hand today. So there's all the genomic, tumor whole genome and germline data, RNA sequencing and methylation data, all stored, raw data stored, all the genomic variants in a database. We've been storing the results from the preclinical drug screening, the high throughput drug screening, information about drugs. Got all the clinical data stored in Lab Matrix for PRISM and now in the Oracle systems for 02. Lots of biospecimens stored in the tumor bank systems. Lots of genomic data in various different databases, information about cancers, the database integration APIs and things, and published data over in EGA. So I'm going to revisit this, I think, in the last slide, which is how do we make it easier to integrate and access all of that information that we've got at our fingertips. I think that's part of the motivation for this talk today is we've got all this data, how do we get it in your hands safely, securely, to be able to analyze that, to do your research as well. Many of you have probably seen me show this. I'm not going to belabor the point. But with Andrew Stone's help, we tried to work out how all these different systems that we've been building interoperate and connect with each other so that we can track data from a patient who receives a diagnosis in the hospital through to enrolling onto the study, with collecting all this clinical data, through the analysis of the genomic data over here with these analysis pipelines to where the data is stored, how it is shared through these different data sharing portals, which algorithms we develop to analyze it, how that data is stored in a database, how all this information feeds into a knowledge base that's live and tells us information about patients and genes and mutations and diseases and trials and all sorts of stuff. So a lot of that data then flows quite as quickly as we can through a molecular curation process where our curation scientists interpret that data, through to clinical curation, where we try and work out what the treatment recommendations need to be, prepare the molecular tumor board presentations. Increasingly, this is done by our precision oncology fellows, back through the molecular tumor board environment, through to various different reports, and back into the hospital. So when we first did this diagram a couple of years ago, there was lots of red and orange because these systems weren't online. But now that we've just launched 02, it's great to see so many systems now with the green traffic light, but there's still plenty to do. So given what I've just told you, a lot of information, right? I've told you we've got lots of data. We've got lots of challenges with sharing that data, lots of need to develop better systems. So we really think about zerochildhoodcancer.cloud as a solution for tackling some of those challenges. So this next part of the talk, I'm going to tell you a bit more about that. So some of the goals for this project really is the main goal is to create a web portal to allow different types of researchers to access different types of data securely, safely, and consistent with the patient's consent. We hear lots of people wanting to access this data, and we have to keep framing that desire to access that data. Is it consistent with the patient consent? If it is, great. So there's several challenges that we need to overcome to build this thing. Security and risk. It's fair to say that we've got a few systems now in front of the firewall, but before that, the safest place for these systems containing this sensitive data was behind the firewall, but that prevents access to you all. Opening those things online opens up significant risks that we've been working closely with Elaine and the TSG group to understand and handle that risk. And we now have a couple of systems in front of the firewall. As I said, consent. Everything we do for data sharing must first and foremost satisfy that patient consent. We realize that lots of people want to access different types of data and have different levels of skills, levels of understanding of the sensitivity of the data, different R programming skills, or lots of different skills. And then ultimately there's the governance question that Xero is responsible for the data and security of it. And as you've seen, lots of data breaches on the front page of Sydney Morning Herald recently. We really don't want CCI or Xero to be on that front page. So when we get this right, not if, we'll open this data up as a broad opportunity for research to improve outcomes in childhood cancer. Some of you will know that our peers have developed the St. Jude Cloud or the Kids First Data Resource Consortium. I'm going to show you a screenshot of that later if you haven't seen it. But I would like to point out they will have teams of over 100, many of whom are working on these resources. So we're getting there. So some of the systems or the components of the cloud that we've already built, Zero Dash, I'm going to show you about that. We've already got the database behind the scenes that has all of the information. Some users may have the right knowledge to be able to access that database. We've built them MTB notes that I'm going to tell you about. Others want to browse the genomic data using the IGV tool. Others want to browse the data using, look one gene at a time using tools like Genome Paint or Protein Paint. Mustafa and the team has built a tool to help look at the genomic data and see the breakpoint in the reads, and then that feeds nicely into Primerdyne so that our MRD group can design assays that are very, very patient specific and span that specific breakpoint. Built Biobank Explorer to try and make it easier to find biospecimens such as longitudinal plasma samples. Sam's been building a patient timeline view that I'll show you later. And Sam's also built an R-Shiny app called Crux, which helps you analyze cancer cohort data. So there's many components that are here. They're all at the moment fairly separate. And the vision is to be able to log in to zerochildhoodcancer.cloud with your username, your same email address, and start to see different components of these solutions. We haven't built that piece yet, so that's why I've been kind of reluctant to talk to you all about it. But these are the individual components, and that's the vision, is to enable that sort of consistent login experience, and then you can access this data securely. So if I go a bit deeper on Zero Dash, I think the first, probably not surprise to most of you, is that one of the major bottlenecks in precision medicine is curating the molecular changes. When we sequence a tumor, we typically find about 1,000 somatic mutations in that tumor, sometimes more than 1 million. And to determine which of these genetic changes is important, our team has to look at a whole dozens of different websites to see if it's an oncogene, if it's been seen before, what Barsome predicts it might be, what PubMed knows about it. And so doing this process for each patient is part of the difference of doing precision medicine as opposed to sort of discovery cohort analysis. And so currently for PRISM, the collective team takes about 7 weeks median from the patient enrollment. We've done this as fast as 8 days. And currently the hands-on time for curation and preparing the molecular tumor boards and reporting is about 22 hours. And our goal is to reduce this really down to 3 to 4 weeks with a 70% reduction in hands-on time. And one of the ways to really speed this up is through building a better curation system. Partners, this is the first time we've actually partnered with software, a proper software engineering company, Secure Agility, and We Discover is a user experience design agency to help understand this process and design it out and model it. And lots of the team have been working on this project to build it. So this is the homepage of Zero Dash. We've given access to this system to those that are involved in the curation of each case to date. So you can log in with your credentials. And when you log in, you see a dashboard of all of the open cases. You can start to see things like this first patient is from 02, and they've already completed their fast track rapid analysis of the glioneural tumor. And we're still undergoing the high risk curation process for this particular one. These are the expedited ones, and you can start to see some of the older PRISM patients in here too. So when the curation team logs in, they're greeted with this, and you can tell which curators are assigned to which case, which is really useful. We start to present the quality control data in a very streamlined way. We need to check QC first, so it passes the contamination check and the C&V analysis check. And you can start to see these genome-wide CIRCOS plots, which show us different features of the tumor. I think our team probably gets a bit affronted the first time they see a CIRCOS plot, but as you learn to understand how to read them, they give really interesting insights into the tumor. You can already tell, once you've seen a few of these, you're suspecting this is an osteosarcoma because of the degree of structural variant complexity here. Once we go to the somatic single nucleotide changes, we've built a machine learning tool called Helium, which tries to reorder the way these variants are ordered so that the ones at the top are the ones that are most likely to be interesting. Here it's a well-established P53 variant that the team has classified as a C5 pathogenic. Turns out this one was actually a germline inherited change that became loss of heterozygosity in the tumor. That's why it's in this list as well. I'm not going to geek out on the genetics here, but this is, really interesting, really useful information for the team to know at their fingertips. You can start to see how the molecular curation team here, Paulette, has written a comment about why this variant is pathogenic with links out to different supporting information. Similarly, for structural variants, these types of mutations that either delete 1 or 2 exons of a gene or fuse 2 different genes together. You can start to see here a deletion which caused an ATRX disruption that the team is classified as C5 pathogenic as well. And this has been classified as reportable because it helps support the diagnosis of this patient. We can zoom in and see in more detail the remarkable amount of genomic damage that has occurred to this particular locus in this chromosome, yet despite all of these structural variant breakpoints, almost the whole of ATRX remains intact, somehow preserving it is the driver gene in this case. I'm not going to geek out on structural variant complexity either, but there's a whole talk in understanding why pediatric cancers often have such a large amount of damage when you look at structural variants and what sort of prognostic impact that might have. Cytogenetics, we try to translate that whole genome sequencing data so that we can understand the cytogenetic changes, which are usually larger scale arm level changes. And once you've gone through that process, you end up seeing a rapid summary of the molecular changes that the team has tagged as being reportable. And you can add comments here if you need to. And actually, this is an old screenshot. Now there's a button there that says reports. And when you click that button, you end up with a dynamically generated semi-automated molecular report, which brings in all the relevant information from the clinical data systems and puts all of those molecular changes into the report with a minimum of fuss. If there's a germline finding, like there was in this case, then the germline team are brought in to sign that report digitally, and then the precision oncologist and the curation scientists get to sign the report and issue it straight from the system. That's much better than the old solution of mail merges and sending Word templates all around and whatnot. So that is, we call this a semi-automated molecular report, and we intend to define one of these that might be nuancedly different for each disease stream. And this is one way of really speeding up the bottleneck, delivering 0-2. There'll also be a molecular therapeutic report from the same system, and that will handle information like the treatment recommendations that come from those changes if the patient has a high-risk tumor, with the right people signing off the right information at the right time. So we're really proud of that. What's next? First half of this year is we need to then streamline how the clinical curation happens, that process of going from molecular changes to recommendations, recommended treatments, recommended trials, recommended papers. We have worked hard on building an MTB presentation layer so that the team no longer has to make PowerPoint files. They can just hit the present button and show these cases in the MTB meeting. We hope that'll streamline a lot. And part of that streamlining is reusing those pathway diagrams or Kaplan-Meier plots or pieces of evidence that we collect when we want to convince the clinicians that this is a really important change that we're talking about. More reporting. What's next, but not really on the timeline yet, is how we extend the analysis to allow the interpretation of multiple tumors. So if a tumor's relapsed, we want to have a more streamlined way of looking at what has changed. We want to have trio-aware analysis so that if mum and dad are also sequenced, we know which genetic changes the child has inherited or picked up de novo. We'll want to integrate this with our liquid biopsy data. And we are working with the MRD team already to work out how we can use whole genome sequencing in their workflow to identify patient-specific genetic changes to those sequences and to design a qPCR assay. So that's pretty exciting. The other more mature piece of the Zero Childhood Cancer.cloud is currently called MTB Notes, because it started off by Alan and the curation team mining the MTB reports that are created and signed off by the Precision Oncology Fellows. So amazingly rich paragraphs of text that link genetic changes to treatments and trials and papers. But over time, it actually needs to change its scope to become a more general knowledge base, because now that we've curated so many cases in Zero, you can now look at your favorite gene, see a bit of information about it, the distribution of mutations in that gene, where the mutations actually fall along that gene. Great information for researchers, of course. And you can start to see what we did in whether we thought, each of these is a different patient where we've reported a different P53 variant, universally not targetable, but often with a comment. And that comment of it in and of itself is useful for speeding up the reporting process. Yeah, you can start to see the actual comments, you can start to reuse them, you can start to see the links to the references. So this is a huge productivity boost. What Alan has just added is a disease page. So for each disease stream in Zero, there'll be a page where we can access not only the gene list, which is what we are using to interpret that particular case, but you can start to see the most frequent diagnoses, information about age, tumor mutation burden, genes that were reportable, really, really interesting information. So there's already a page for the CNS disease group. There'll be one for neuroblastoma. There'll be one for leukemia eventually. So this is really powerful tool for browsing the data. Oop. So if I pivot now to the new things that are new opportunities, largely unfunded, there's three components here. So I guess what I've told you about, I've told you you've got lots of data, we're thinking about ways of building systems to help liberate that data. We've shared those systems with people that are, core to the curation and interpretation of the cases in Zero, and thinking about how we get broader access to those systems through the cloud. What I want to focus on now is three new opportunities. One's the Zero Data Commons, which is going to be a portal for browsing the raw genomic data. A Precision Medicine Data Lake, which is a brand new database type structure to browse and integrate these data. And finally, how we might harness clinical data directly from the electronic medical record. Okay. So if we think about sharing raw genomic data for a moment, we can start by imagining that Zero has developed a unique national, perhaps even global data asset, but researchers can't currently see how much data we have, what type of data there is, and that makes it pretty hard for them to know what they can even request access to. So I've told you earlier on in the talk that there is a data access procedure, and Biljana and the team on that email address will help you work out what data is available. But we recognize that that's a poor substitute for you seeing it, browsing it, and being able to actually feel that data to know what you would like to request access to. So we're aware of that challenge. The data access procedure then uses an offline form, which is a bit labor intensive for the user to write and the reviewers to review. So that could be streamlined. The comp bio team's process of delivering the data to those users is fairly labor intensive too. And we're starting to see international data sharing initiatives use various data standards, but they all use slightly different standards or different definitions of diagnoses. So you take, for example, find me all DIPG patients at St. Jude or CHOP or ourselves, and we all might define that specific diagnosis differently. So it's kind of hard to run a computational query and get an exhaustive list of all of those specific patients. And that just gets more complex the more specific your query needs to be. So what is emerging is the concept of data federation. That's becoming the de facto standard for sharing genomic data. So if that's a new term for you, that kind of means that each participant that wants to share their data builds a system. And these systems, so the data stays with that data owner. And then the data federation allows queries to be run so that you can find out what data lives in those different systems. And then you can request access to that data. As opposed to saying, hey, if you want data, well, you have to upload it all to one big database. And then who owns the database and who's responsible for maintaining it? These are difficult questions with the older solution. So I mentioned this earlier, but two of the biggest groups, a bit like our group, but much larger, have built the Kids First Data Resource Consortium and the St. Jude Cloud. These groups have taken it pretty seriously as to how you can access these data. I already commented that they have groups of over 100 engineers working on these solutions, but it is definitely our intention, more than an intention, as I'll show you, to build a data sharing portal so that you can browse and see how many patient data are available for the Zero data. So a potential solution is what we're calling the Zero Data Commons. That's a new term for you, kind of the idea of a commons being a meeting place. There are various data commons platforms now, from the genomic data commons to others that I've just shown you. And so essentially this will be a web-based platform which allows researchers to browse the catalog of data, find their data of interest using some reasonably sophisticated filters, such as find me all the patients with a specific diagnosis and a specific age and a specific genetic change and a specific survival data. That same example I gave you earlier. It's important that a researcher needs to authenticate and log in to that system, demonstrate that they are indeed a researcher. be able to request access and receive copies of the data via the same platform. We want the platform to be standards compliant to try and tackle that challenge of find me all the brain tumor patients or find me all the DIPG patients. We need to use clinical data models. I know there are a couple of people online that know about clinical data models, so we are also leaning towards this OMUP clinical data model. As we all know, diagnoses can be organized hierarchically. So you might be interested in all brain cancer patients, and someone else might be interested in all medulloblastoma patients, and someone else in the sonic hedgehog subtype of medulloblastoma. So all these diagnoses are actually hierarchical. And so we want to be able to represent that hierarchical organization and make it easier to then find data at different granularities. And so sort of borrow the best features from the other platforms that exist. And once we've solved this problem, we can then federate our data with other data sharing platforms really elegantly. So feels like there's lots of initiatives in this space right now, but this solution solves almost all of those initiatives. Not to get too nerdy, but there's an organization called the Global Alliance for Genomes and Health. It's got lots of international uptake. And one of the projects that it's developed is this Beacon V2 API, which is a standards compliant API for sharing of computer solution for finding and accessing genomic data. I don't want to get deep into how it works, but we are aligning with this standard. These are designs that we've just been updating about what this data commons might start to look like. So I've already shown you some screenshots from Zero Dash, but this is a potential, as I said, this is design, this is not implemented yet, but you can start to see that there are ways to have lots of different filters pop up, and you can start to then drill in and find different patient characteristics which satisfy your question, which then restricts the types of data that you see. You're probably not going to see any of this sort of detail about how the curation happens for when you're browsing this publicly. But that's part of the challenge, right, is getting what data should we be exposing when you are a curator versus when you are a researcher wanting to browse it. So I just want to shout out Mustafa, Camille, Marie, Angela, and Zoe, who've been building this. So we're optimistic that this will have something functional this half year to be able to tackle this. We're also starting to think about how you might browse the cohort, and this is fake data, but start to work out how many different disease diagnoses and different visualizations for how you might interact with that data to help you discover the data that's most relevant for your research question. Getting nerdy for a moment. You'll need to log in as a researcher. And so we need a way to prove or to demonstrate that you are indeed a researcher or a clinician with a current affiliation with an organization. So we're partnering with the Australian Access Federation, which uses this global technology called CI Logon, which will allow you to log in with your Harvard email address or your UNSW email address, and us know that you're an accredited researcher. The other piece of the puzzle is a system called REMS, which allows you to submit data access requests and for us to streamline how we approve those data access requests. Again, standards compliant. And so we've been piloting this as a solution too. Haven't explicitly mentioned that all of this work is in partnership with the Australian Biocommons. There's a big grant called the Human Genome Platform Project. And so we've been working with a national team to evaluate different technologies for each of these pieces of the puzzle. And these are the ones that we've collectively settled on. My intention is for you to not know about any of this complexity behind the scenes. It's simple to log in, simple to browse, simple to access, and that these technologies happen all behind the scenes. So we are aware that many people don't like having lots of different systems to do different pieces of the puzzle. We're trying to make that problem go away by hiding it all behind the scenes appropriately. So if we revisit this page earlier, what's different? Everything I've talked about is sharing zero data, but if we can do sharing of zero data well, then we should also be able to solve this query of sharing zero data, University of Melbourne data, Garvan data, and others using that same collaboration with the Garvan, with the Australian Biocommons. So if we look more closely on the left here, you can see this register beacon button. And this is the idea that if Zero has a beacon that allows us to browse our data, and then Garvin has a beacon, and University of Melbourne has a beacon. And here they are representing the Australian Genomics Health Alliance as well. So they might have 2 beacons. This is QIMR and NCI, etc. So these are the partners that are working on this project with us. And the idea that you could then run the same query across all of these beacons and get a unified result back is something we're trying to solve. So one solution to solve both of these problems. Again, pretty excited that this is working really nicely, will work really nicely. So if I just try and summarize, it could be a bit complicated, but there's a bunch of technologies that we're developing to make it easier to share genomic data. We've talked about building a zero data commons where you might be able to browse the zero data. We have other bits of data too, like PDX models and drug screens and things. So that might be available when you research a browser's this one. And using the same technology, the researcher might browse this biocommons beacon network. And so they might put the same query into this system and get a result that aggregates across all these other different beacons. And then there are a couple of international initiatives emerging too. I've mentioned Minderoo's Federated Cancer Data Bank. I'm not sure how much I'm allowed to say about it yet, but it's certainly going to have some of Zero's data and a couple of other prominent groups in it. And the idea that you might run a query there and access data at multiple sites. And the HOP is an international data sharing initiative just launching out of Europe. So takeaway message there, one technology will hopefully solve our ability to share data domestically, nationally, and internationally. Just keep an eye on the time. I want to leave some time for discussion. So I just want to very quickly say that there is a new data architecture called a data lake. This seems to be emerging as a really innovative data solution for having data that belongs in databases, which are very, very structured, and data that is very unstructured, like heartbeat data streaming off a watch or like a Garmin. There's some challenges for data lakes that I probably won't go into for the sake of time, but there's a new data architecture called a lakehouse, which has the best of both worlds, where you can have structured data and unstructured data all in the same data system and provide a single front door, a single way to securely provide access to that data. And so we think, I'm not aware of any research groups that are implementing this yet, but we think this is an opportunity to bring together all of these different data sources that I showed you right at the start of the talk, put it all underneath a data lake architecture, and work out what types of research questions this solves for us. And working closely with TSG and governance so that we know that we're providing access securely and safely to these data at the right time, in the right way. This is the start of an honours project led by James Bradley. So that's really at the start, but by the end of the year, we'll have assessed whether this is going to be useful piece of technology for data sharing. I'll skip that. I think the last thing is really about clinical data. And if we can do this from the electronic medical record. So I want to leave, Ness, I'll take for 2 or 3 minutes to whip through this and I'll leave some time for questions. So I think we all implicitly know what the core problem is here. So collecting data for clinical research is expensive and error prone and labor intensive and sometimes uses paper-based forms. Major expense of running clinical trials. And the bit that is most frustrating is you have to be very pragmatic. If you can't put questions on the data collection field just in case it's going to be useful in the future, it's very pragmatic. It has to be useful to warrant every single nurse CRA entering that information for every patient at every follow-up, right? Even more, some children will interface with 4 or more studies. Their data will go into a registry, their data will go into a biobank, they'll enroll on 02, they'll go on a treatment trial. And a lot of this information is exactly the same, collected 4 different ways, leading to difficulties. So we think this is a big problem and it can be solved. So just to reiterate that there are different types of data. Some of it is structured. So you can read the name out of a database. Some of it's semi-structured. You're going to have to read a sentence to work out what it means, and others is unstructured. And so there's different solutions for how you might track structured, semi-structured, and unstructured data, feed it into a data lakehouse, clean it all up, simplify it into a simple iPad app, so that your CRA at the site can then select the patient, match to the research studies that are relevant for them, import all this clinical data from the big system, fill in any missing data, because there will be missing data, make sure it's de-identified, and finalize that case, and then push that information out to all these different systems that already exist. Sounds simple. This is kind of the big vision that we're calling Tundra. We were unsuccessful getting this funded through MRFF, so we're still trying to work out how to solve this. But yeah, we're optimistic that we can make some inroads here. We're making inroads in using natural language processing to read that text, so you can pull out the meaning with computers and then have a human verify if that's actually correct. I'm so excited about that. And that's way too detailed to go into, but it's how you might build the model, refine it over time with our expert input, and leverage that data from 0-2 as the gold standard to make these data extraction models really accurate and streamlined. So the end result of getting that right is, instead of all of that manual data collection, we should be able to stream lots of this data straight out of electronic medical records. We believe we need to start at one site, and so that's starting with the Sydney Children's Hospital network, and then move from there to the next site, because each hospital system is different. And to give me optimism that we can do this, we've just launching in about April, the next phase of the Luminesce Alliance, which will bring together myself, Tom Snelling, who's an expert in clinical health informatics, who has a group within the City Children's Hospital network able to do these types of queries, and Natasha Nassar, data linkage expert. So bringing the three of us together to try and tackle this together, not just for kids' cancer, but for children with disease, is going to be really powerful. So I've said a lot, but I think I was trying to go broad, a little bit deep. We've got a very diverse audience. If you take nothing away from this other than we're pretty focused on building systems to get this data in your hands. It's taken some time, but there's, as you hopefully heard, there's been some really essential systems to get right before we can launch 0-2. And so this is a major focus right now. Over the next 6 months, you'll see a new data access policy, a new data dashboard in the form of Zero Data Commons. To make this all work, we have to work out personas of different types of people that want to access the data with different capabilities and skills, work out how to onboard you into these platforms and what training you might require to be given the keys to the data system. And then after that, work building more and more researcher and clinician dashboards to interact with that data and begin to start to stream that data out of the electronic medical record through our partnership with the Luminesce Alliance. So with that, I would like to say thanks again to the group who really make this happen. We're getting to that point where the expertise in the group is clearly outstripping my knowledge of some of the subject matter, which is an awesome place to be. Thank you so much to the funders of CompBio specifically, but the huge effort from the Zero team, especially leadership of Ness and Michelle. Many of you on the line who support the program, and a huge number of supporters who make this happen. So I might stop there and say thank you. And so if there's any questions.
Thank you very much, Mark. We do have a few minutes for questions. So you've heard quite a bit today. That was a very dense piece of work from Mark. Thank you very much. And just to reiterate that we are going through a complete overhaul of the data access policy and principles and processes at the moment. And you will hear more about that when we have that endorsed by the committee and the board. So over to you for questions, either in the chat or feel free to put your hand up. And we just have a comment in the chat. Did you want to say anything to that? No? So we've got great talk. Interesting to hear about all the data engineering problems and solutions that occur behind the scenes. Thank you for that. Any questions for Mark?
Sure, I read the room right, but there's lots going on. Sometimes that needs talking about.
Toby's got a question
Toby.
for you. Toby.
This may be more of a comment. Sorry I was late to the meeting, but great work. New South Wales Health is going to struggle with exactly the same issues, and they've got the clinical trials governance accreditation framework for all hospitals now. They're in the process of putting in, a clinical trials management system, and they're going to face exactly the same issues about clinical data collection. So I suppose my comment is I think this is really nice work. It'll be great to see how it goes, but I think there is scope for this type of work to be used more broadly within clinical trials research in health broadly in Australia and internationally. So it's really nice to sort of see the collaboration with Natasha Nassar and Tom Snelling, and I think it would be great to see how far this goes. So great job, well done.
Thanks so much, Toby. I think there's dragons to be slayed ahead, right? The New South Wales eHealth has just kind of changed the landscape a little bit with how you access the Sydney Children's Hospital EMR. So that's a bit less streamlined than it was. So we're trying to work through that. But I totally agree with you. We're hoping these are exemplar solutions to problems that we know are widespread. Trying to balance, do you go national? Do we get all partners involved and start conversations with the CIO of every hospital? That seems like a hard place to start. So that's why we're optimistic about making some traction at Sydney Kids and seeing where we can scale up from there.
Mark, I might just butt in with a quick comment. Thank you. That was a really great summary of a lot of complexity there. And just to note that I think, the approach you presented is great because, as you've acknowledged, there's a lot of different use cases here. There's a huge diversity in people wanting to use the data, and both from a operational perspective within the program, and then obviously the many different use cases at the end. So I don't think we'll see a one-stop shop that manages to tick off all of those different use cases, whether it's clinical data, the imaging, the genomics data, and so on, because there just isn't that case. So I think the approach you presented today is great with that multiple layers of tackling the different problems there and acknowledging that each one of these takes time and has complexity and has to get the data access right to ensure that it's protected but then shared appropriately. So no, really appreciate you taking the time to present that vision today.
Thank you, Kim, and looking forward to learning more about OMOP with you and working with you on the Federated Cancer Data Bank mission.
Absolutely. Yeah. Cheers.
So we have a couple of comments in the chat. Jordan says, great talk, Mark. Love MTB notes. Fantastic work. Makes her job easier. Jessica has asked, in the future, will any of those automated reports be transformed into a family-friendly form?
Yeah, great question, Jessica. I know Eliza's on the line who would love to see that happen. And we see the templates from the Australian Genomics Health Alliance. So if the germline team would like to make it so, and we would be very keen to see if we can make the technology support that. But I think I'm not the decision maker on that one. I'd love to facilitate it if the germline team would want to drive it.
And Maria Kirby says, thanks, Mark. Just need to nail the problem of multiple EMR data custodians, especially given the recent public breaches. Right. Very much looking forward to seeing this evolve.
Thanks, Maria. And I skipped over an important point there, which was that this data lake, I've talked about a zero data lake, but we're proposing a data lake within each hospital environment so that they control it. They're only leaking data out of that environment that we have consent for in the zero system or the Inform 2 system. So we hope that helps de-risk the process. It would be much riskier to say, hey, Zero's built a data lake. We'd like to stream all of your health data into it. And so we think that's a way of de-risking this. But you're absolutely right, Maria. Each hospital, different custodians, different initiatives in each state. Dragons ahead to try and make progress in this.
Thank you, everyone. We are out of time. So thank you very much again, Mark. Thank you to all of you for joining us. Our March Zero seminar will be a special seminar with 2 invited international speakers who are collaborators who will be here in Sydney on the Thursday, the 30th of March. For those of you in Sydney, there'll be an in-person seminar at City Children's Hospital between 12 and 1. And of course, we will have that virtual online so that all of you can also join that. And as always, we welcome speaker nominations from you. So please do reach out. We'd love to hear from you. But for now, thank you everybody for joining us today. Take care.